Just picking up where we left off in class, we were talking about single unit recording. We had an example here of showing an animal or teaching or training an animal on a very specific or complex task, and then being able to record single neurons or batches of neurons. And so we talked about being able to do intracellular versus extracellular recording. If you do intracellular recording, you can see what that specific neuron, when that specific neuron is firing. But of course, a lot of the single cell recordings are done with within extracellular fluid, and so it's really difficult sometimes to pinpoint exactly one specific cell. And if we have, for example, a sort of inability to tell the difference between the cells, and we're actually capturing cells around this particular neuron that we're interested in, that's not going to be a single cell recording. So, even if we're able to get information from 3 neighboring cells, that is good, but it's not what we want if we're doing a single cell recording. And so it's always important to consider that neurons are always active. They're always communicating with one another. They have different baseline firing rates. Different kinds of behaviors are elicited by different kinds of firings and activations, and that might be an increase relative to baseline, which is different from cell to cell. Just because we see a decrease in firing doesn't mean that that's necessarily a bad thing. It doesn't necessarily mean it's a good thing. And sometimes a decrease in firing can actually elicit behaviors if we're talking about inhibitory neurotransmitters or inhibitory neurons. And so it can get really complicated when you're doing single unit recordings. But the really interesting thing is that you can really isolate different information for these particular kinds of cells, and get some really good readings depending on the nature of the study. So when we talk about the single unit recordings and also the possibility of stimulating particular neurons, we can also do this. We've seen this also done in humans when we talked about Dr. Penfield's being able to do surgery on patients with epilepsy who have basically their brain exposed, and he's able to stimulate different parts of the brain during neurosurgery to know, okay, let's map this portion out and then not operate on something that's vital to function. But when Dr. Penfield did this and was able to stimulate different portions of the brain and map out that sort of homunculus of sensory and motor cortex or cortices, he really put together that you can stimulate different brain areas and then observe what kind of behaviors come from that stimulation. When you look at animals, for example, if you were to take a rat and put electrodes on the lateral hypothalamus, whenever you stimulate that portion of the brain, the rats will actually engage in eating behavior, and they will even self-stimulate this particular area of the brain to stimulate hunger. And so we know that very specific neural circuits, especially those that contain dopaminergic pathways, are related to pleasure and reward. And so we know that it's likely that we can actually recreate this and have animals who are actually experiencing a pleasurable sensation with stimulation of this particular area of the brain, and engage in things that are also pleasant, like eating behaviors. And so this can give us a lot of information about recreating neuronal firing to see, okay, what is this particular area of the brain or patch of neurons responsible for. But another thing that we can do is actually observe electrical activity So we're going to talk about how we're looking at electricity in the brain, electricity in the nervous system, and we're also going to talk about stimulation again later on. But just a little bit of history here. Hans Berger in the early 20th century was the first person to be able to record electrical activity through an electroencephalogram, which electroencephalogram allows us, EEG allows us to record the electrical activity using electrodes on the skull and capture graded potentials of neurons to identify what neurons are active. Now, these are not action potentials, and we're going to talk about that difference here in just a minute. But let's just be very clear that this is not actually capturing action potentials, but graded potentials. So, an encephalogram or EEG is a recording of the brain potentials or brain waves. Here you can see that there's a person with a cap, so we can do it with a cap, or we can do electrodes directly on the scalp. These are actually on the scalp, but they're sort of held in place with the cap. It's a little bit easier to be able to, it's a little bit faster to be able to get these caps on and off. But if you notice that there are different electrodes throughout the over the scalp, and we can get many electrodes in. You can get 120 electrodes in, for example, or you can go smaller and have maybe 32 electrodes. But these are, or 64 electrodes, just depending on how many particular electrodes you have. And you need to be able to ground these and get a baseline. So typically we also have an electrode on the ear. We can also obtain eye movements, which is a different process, but that's very helpful when it comes to sleep, because when you know what kind of brain waves you're experiencing, you can actually identify what stage of sleep a person is in based off of brain waves, eye movement, and muscle tone. So these are things that can help identify things like epilepsy, sleep, lots of different things that it's used for medically, but also it's used heavily in research to identify brain activity based off of particular stimuli. And we'll talk about that in just a minute. So when we look at ways that we can see brain activity, okay, so EEGs in electroencephalography really allows us to be able to see what activity is going on temporally. So EEG is really great for being able to identify a point in time where the brain is reacting. It's not great spatially. So being able to identify exactly where the firing is taking place, it's a little bit more problematic, and we'll talk about that here in just a few minutes. But what's really helpful about EEG is that it allows us to be able to see synchrony across areas of the brain. So in a typical brain that's developmentally appropriate, we typically see desynchrony on an alert brain. But when we see a person who's having epilepsy or having seizures, we actually see synchronous activity, and we see what we kind of refer to as overfiring or firing that is really not supposed to happen at that time. And so here you can actually see this would be a typical EEG. Here, I'm trying to get my pencil to work here. Here is a normal EEG. Or you can see that, there are different waves taken from different electrodes that are typical patterns. And then you can see the desynchrony. They're not synchronous in terms of activity. Whereas here, a person's experiencing a partial seizure, so you can see synchronous activity here, but normal activity here, whereas a generalized seizure is one that kind of covers the both hemispheres of the brain and is across the whole brain. And here you can actually see this particular pattern. This would be an example potentially of a much more serious seizure, which we'll talk about here in just a second. So there's three basic categories, and other people will sort of categorize them slightly different, but we have these generalized seizures, which we see activity across the brain. And then we might see localized seizures where we see perhaps a focal point where it starts in a particular location and then moves across the brain. And then sometimes you can see that a particular seizure is only happening in one small portion of the brain. So this is kind of how we categorize based off of are we seeing whole brain activity? Are we seeing a portion of the brain? And so here's an example. You can see on these EEGs that these are generalized categorized seizures. So this first one is a tonic-clonic seizure, also sometimes referred to as a grand mal seizure, grand being a French word for big. And so this is a seizure where you typically see the muscles are going to contract, and then you're going to see potentially the person convulse. And this is what most people consider when they think about a seizure, that a person is sort of convulsing on the floor. And this is usually followed by a deep sleep or confusion, and then sleep. The person gets very tired. And here you can actually see, so this is an example if we were to have electrodes on, the left frontal, the right frontal, right temporal, right occipital. Here you can see each point. Before the seizure, you see a typical activity desynchronized. And then during the seizure, you see, of course, synchronous activity. And then after the seizure, you typically see waves that are associated more so with rest, sleep. And so this would be a generalized, very serious seizure where the person is experiencing likely muscle contractions, and then once they're unconscious, and then once they regain consciousness, they're very confused, and they don't feel well. Another type of generalized seizure here, this simple partial seizure, and you're going to see very, very brief periods of synchrony, so maybe 5 to 15 seconds. Here you're going to see the brief period here. We don't see the kind of convulsions. You don't. Actually, the person really, the only kind of motor activity you might see is potentially some repetitive behavior, but typically they sort of pause. They really, really are quiet. Their eyes might move, but they sort of lose their focus. They lose their ability to respond. And they typically don't remember this. And a lot of times, and these are also referred to as absent seizures, a lot of times what a person might do is they might say, they might call, especially in a child, the child might just kind of go quiet for a second, and you might call their name and they don't respond. And it might only be just a few seconds, and then you call their name a couple more times and they don't respond, and then you call them again, and then they kind of break out of it. And typically what parents will do is if it feels weird or seems kind of odd that the child is sort of ignoring them at different brief points throughout the day, they'll start recording these, and then you can give it to your doctor and say, hey, there's something going on here, and it might be suspected for simple partial seizures, and then the child would then be evaluated with an EEG over the course of, depending on a certain amount of time, might be 2 days, or might be a day, it might be several hours, or it might just be one EEG, depending on this is the doctor and this is the suspected case, and then that brain activity is monitored to determine, is that person experiencing seizures. So we don't typically see the big convulsions. We might see a little bit of repetitive movement, maybe rocking, something like that. But generally the person stops and maybe stares into space, loses a focus with their eyes. You don't really see them really do much other than be really quiet for just a really, really brief few seconds. Another category of seizures are the complex partial seizures. They do not involve the entire brain, as you can see here. This would be an example where you can see that portions of the brain might be involved. And you might see, for example, a focal point, and that might be, let's just say that might be focal point right here, and this is where you might only see the seizure activity really happening. Many people experience some kind of aura, which is like a sensation. It might be a bright light, or it might be a sound, or it might be something that precedes a seizure, where it's almost like a really quick warning that the person is going to experience a seizure. And it's just enough time for them to maybe sit down, or know, okay, I need to get into a safe space because I might have a seizure. Now, if you were to look at some of these intracranial readings, here you can actually see, in this case, you can actually see what it would look like. This is similar to the other picture I was showing. So these are 3 points in the brain. If we were to do intracranial recordings where we actually have electrodes directly on the brain, you can see where this synchronous activity occurs here, and can tell us that, it likely is whole brain if these all 3 specific areas on the right and left hemispheres are all demonstrating the same activity. So that would be a synchronous activity, a generalized seizure across the brain. So here's an example of electrode placement. Actually, so this is something that might happen right before a brain surgery. If a person has really severe epilepsy that's not being able to be treated by medication, they'll sort of map out an area here and then place electrodes directly on the brain. So this is not the scalp. The scalp, the skull is removed here, and the brain is exposed. And so these electrodes are placed directly on the brain. Sometimes we are able to get some really, really interesting experiments done, where we're able to get these multi-electrode stimulation and recording experiments through people who are already having brain surgery for epilepsy and then agreed to take part into further experiments. However, typically we see very, very low ends, so that means that the subject number is really low because you might only get 4 or 6 people to participate. But this is something that allows us to get brain surface recordings, and it's better than scalp recordings, but it also is limited to people who are experiencing seizures and epilepsy, and so it might not be generalizable to everyone else. But very interesting ability to be able to get more information on things like dreaming and sleep. And we've learned, for example, that people will, when you place these electrodes and you ask them information about their day, and then you have them sleep, and of course they have these electrodes placed during sleep as well, and they might have them for 24 hours. And over the course of the night, or even more than a day, over the course of the night, you can actually see the same patterns during the night as we saw during the day, of them going over some of the things that they did during the day, which gives us some information that when you sleep at night, when you go to bed and you go to sleep, that you're actually sort of recreating your day, and you're thinking back to all of the memories and things that are happening to you throughout the day. And that's likely where the brain decides to throw away memories or keep them. And so we are only able to get this information through this kind of recording. So it's a really helpful way to gather information. So here's a couple of different versions. So here's a cat. You can see basically these electrodes are on the scalp, and you take like a little stick and you make it so that you can connect to the scalp, just to kind of move away a little bit of the dead skin. And then you have a gel which you enter in here to allow for better conduction. Here is an example of not a cap. This is actually like a net here. And then if you go to a doctor, you might actually get separate electrode placements. This is harder to put on than this. And then obviously you can see there's more connections here. So you can have like a 32 or 64, 120 plus cap that you can get recordings from each sort of part of the brain. So when we look at what we are actually getting in the EEG, so here would be sort of the surface of the cortex. What we're looking at is pyramidal cells. And if you can see here, look at the dendrites and the connections on the pyramidal cell. We are looking for graded potentials through these pyramidal cells. So we're not looking at action potentials. We're looking at those postsynaptic potentials on the dendrites. These are graded potentials. So here's the surface of the scalp, and the electrode is here, and we're looking at this information here, both afferent and efferent axons, to see which ones are they give us a positive wave versus a negative wave, if they're afferent versus an efferent, and that will give us some information on what kind of graded potential activation we have. But remember, these are not actually action potentials. So it's easier to get information from the perimeter cells because they are perpendicular to the cortical surface, and it's much harder to get information from the sulci because these are parallel, and these electrodes are picking up that perpendicular activity here. So it's much harder to get sulci. It's much easier to get gyri information just because of the nature of the pyramidal cells. Here's some examples of the brain waves, and these might be one from one particular electrode or more synchronous activity depending on what kind of brain waves, what kind of experiment you're interested in. For example, we can look at specific brain waves during sleep. We can look at specific brain waves during drowsiness, during awakefulness. So this might be beta waves. This is where a person is actually experiencing some, they're concentrating, they're thinking. We see alpha waves when a person's more relaxed or when they become more drowsy. When they're asleep, we usually see a combination of waves. So we might see some alpha waves. When you're in REM sleep, which is rapid eye movement, your dream state, it actually looks a lot more like this. It looks more like the sort of sawtooth beta waves. And we'll talk more about sleep when we get to that section, but just kind of bringing in some information here. Delta waves are what you see during deep sleep, and these are all measured in hertz. This is these big fat waves that indicate that you're in a deep state of sleep, and then when you wake from these particular this stage, then you typically are very confused and you're not really sure. Here you can see a coma with very large waves, but these are ones that are much harder to predict, and this person might not have very active brain cells because they are in a coma. So when we're interested in measuring potentials, so what we do is we start with the brain waves or the electrical activity, the graded potentials from EEG, and we isolate ERPs or event-related potentials based off of an event or some stimuli that we present to the person. So this would be example of different trials, and then you're going to take that information and you're going to repeat it over and over and over again. You're going to repeat that stimuli over and over and over again because you're trying to capture a pattern of activity of the brain's response to that specific thing. Okay, so you would take each of these trials and then you're going to consolidate them together, and you're going to try to isolate this event-related potential over a period of time. So we know that there's a delay in response to different kinds of stimuli, and we're interested in measuring that slight delay and looking at the afferent connections, axons, to see what kind of information that we can consolidate. And so what are ERPs? Event-related potentials are those potential shifts that's caused by a discrete, unique stimuli. So in the case of, for example, auditory stimuli, so we can present an auditory stimulus here and actually measure over the period of, let's say, 150, 200 milliseconds, what kinds of reactions that we're seeing in the brain. And then we can isolate the different parts and say, okay, what specific wave are we interested in? What reaction are we interested in? In this case, we can actually identify things like hearing impairment based off of that activity. So we can look at this over time, and we can plot this, basically plot the scalp topography, look at the examined changes over time. So here, this is right after the onset of the stimulus. This is where we're starting to see some activity here. Here you can actually isolate that information based off of this chart here, and then identify where the brain, at what point in time the brain was responding to that particular stimulus. Now, remember the EEG is able to, we're able to calculate these ERPs and identify these parts after, tons of trials over and over again. But EEG is really good at temporal information, giving us this very specific information over time, over a series of milliseconds. But spatially, it's not very good. So even though we've got an idea of this activity here, 150 milliseconds after the stimulus was presented, we know that there's activity in this particular region, but knowing exactly where it's occurring is a lot more difficult. So we're trying to get information from those graded potentials. And so remember, we're looking at IPSPs and EPSPs. So IPSPs are inhibitory postsynaptic potentials and excitatory postsynaptic potentials, EPSPs. The positive and negative doesn't necessarily mean an increase or decrease in activity. We're looking specifically at afferent versus efferent activities. And so remember, if a particular area of the brain is inhibitory, that is just as important for us to know if something is excitatory. So the positive versus negative voltage doesn't necessarily mean no activity versus activity. It's specifically looking at EPSPs and IPSPs, afferent versus efferent input. And then what you're doing is essentially over this period of time and comparing conditions, you're going to compare electrodes to each other, and you're going to basically look at the differences between all of these electrodes, and then that is going to give us an information on potentially the estimated location in the brain. Now, what you want to do, of course, in best case scenario, is actually have a map of the person's brain through something like MRI. So you're not having to guess based off of somebody else's average brain. You're actually looking at the person's brain and you're plotting it onto the person's own brain. This isn't always, you're not always able to do this. So sometimes we have to get, this is like a generic brain, and we're hopefully plotting some activity that's located in the brain. However, we're always going to run into issues with where are we in terms of location? Are we more subcortical? Are we on the surface? Being able to differentiate exactly where in the brain we are is much harder with EEG because we're only taking scalp recordings and we're only able to go so deep into the cortex. And so it might be really difficult for us to measure exactly what kinds of signals we're getting from where. Temporally, fantastic. We can absolutely get the reaction in the brain, but where exactly is it happening? Is it more on the surface? Is it more subcortical? Being able to map that out, that tomography, is much, much harder in EEG. So real quick, just kind of moving on to a different type of a methodology. MEG is all about magnets and being able to since we're able to, our neurons are able to generate electrical activity, that is also going to generate differences in magnetic field. And so EEG is sensitive to all sources, but it's most sensitive to those radial sources that are perpendicular to the scalp surface, whereas MEG is only sensitive to tangential sources, meaning the parallel. So if we could put them together, that would be a really great way to see whole brain, but it's also really difficult to combine methodologies. So EEG really great at these perpendicular sources as to the scalp, and then MEG really good at parallel sources, and it is, of course, looking at the magnetic field. Now, MEG is really, really great. It's really interesting, but it's also extremely expensive. So this is an example of one of the machines that you would use. You would actually compare the cost to fMRI, which can get very expensive in the millions of dollars. The analysis that you do is very similar to EEG in terms of mapping and tomography. And it's even easier to do things like source estimation. Where in the brain are we talking? Because we don't necessarily have to worry about the skull, because the magnetic signal is not, there's no interference with the skull. Whereas electrical activity, you're going to have some interference with the skull, and that's going to change how likely you are able to actually identify specifically subcortically where you are. Now, TMS, or transmagnetic stimulation, sometimes TCMS, transcranial magnetic stimulation. This is looking at electrical current that is produced through these coils here, and you can actually stimulate specific areas of the brain, and we call it sort of temporary lesions. So it's like, making that particular area inactive or making it active. So you could stimulate the neurons, for example, in the motor cortex, and make a person raise their hand or move their arm. You can also temporarily inactivate particular regions. Let's say you inactivate the same region, and that is going to then, by overstimulating them, and that will sort of make you lose the ability to use your arm very, very, very temporarily. And so it's a really interesting technique that can be used at changes of activity in the brain, and a lot of times it's used with other neuroimaging methods. For example, you might take an MRI structural image of the person's brain and then try to stimulate a very specific area. Because you have a picture of their own brain, you're able to actually really get a good picture of what's happening. TDCS is transcranial direct current stimulation. So this is using magnets, right? So this is transcranial magnetic stimulation. This is transcranial direct current stimulation. It's kind of the electrical equivalent of TCMS or TMS. And again, it's just using wands to be able to stimulate very specific areas of the brain. And it's something that can be used for certain kinds of conditions. We also see transcranial magnetic stimulation, which is not as invasive as it sounds. Can be used for things like bipolar disorder and depression. And there's actually a researcher in Charleston who is using this experimentally at MUSC in Charleston, and getting some really interesting results, some really good results. The last methodology I want to cover is optogenetics. I just want to cover this very, very briefly. I've got a nice YouTube video here for you guys to take a look for more information. So basically what you're doing is you're using light to control the brains, and these are through to control the brain, and this is through light activated proteins. So you can actually do surgery excuse me, on animals, because we have learned that different kinds of bacteria, different kinds of proteins are able to produce proteins that are reactive to light. And so if you're able to actually do experimentally, like for example using mice or rats, you can actually knock out or include genes that are like that deliver light-sensitive proteins to target cells, and you can illuminate the cells and actually see what they do. So here, like I said, we found that some algae and bacteria do produce light-sensitive proteins. These are called opsins. We have opsins that are in our eyes because these are light-sensitive receptors in our eyes. But this is something that you can actually do. It's an invasive procedure that you can do in animals to actually make them produce these opsins. So for example, you can use a channel rhodopsin, which is going to respond to blue light, and it does so by allowing sodium ions to enter the cell, which is going to depolarize the cell. And so you can look specifically at different kinds of neurotransmitters and different kinds of pathways. Another kind of molecule you can use is halorhodopsin, which responds to yellow light, and that allows chloride to enter the cell and is hyperpolarizing. So you can use channel rhodopsin to look at depolarization and halorhodopsin to look at hyperpolarization. Here's an example of a rat that's gone through a procedure. So you essentially you put this implant in, and then you can stimulate a particular class of cells that makes the opsin, and depending on if you use like long or short pulses of light, you can actually see you can see spontaneous behaviors. And so one way that they do this is actually scientists will actually arrange for only certain types of neurons to make the opsin through genetic methods. We've talked about things like CRISPR and stuff like that. And so the idea is that you're going to see only those cells that's going to respond to the light stimulation. And so it's a really interesting technique. Obviously, this is not something that you can use on humans. This is something that is used in animals, in animal work. But it's a really interesting technique, and not to sound corny, but sheds more light on what the brain can do. This is the end of our methodology discussion, and this is in relation to one of your written assignments. So hopefully this will give you some more information to be able to complete that assignment. Stay safe, and we'll see you next class.